Okay, so hello everyone, my name is Ghazal Aghagoli. I'm a rising senior at Brown University studying neuroscience, and this summer I focused on the role of Wnt signaling in hepatobiliary differentiation and liver outgrowth in zebrafish. Now the reason why I was interested in Wnt signaling is because alterations in Wnt signaling has been found in many cases of hepatocellular carcinoma as well as cholangiocarcinoma, which shows that the Wnt ligand actually acts on multiple different cell types in order to induce carcinogenesis. Now, I was interested in studying this in zebrafish as a model organism because developmental signaling pathways between zebrafish and humans are pretty much conserved. And so, with that said, I'm going to talk a little bit about what canonical Wnt signaling is. Now, I won't go too much into the mechanism, but what I really want to highlight is the fact that in the presence of Wnt, you end up having accumulation of beta-catenin in the cell cytoplasm, which translocates into the nucleus and actually leads to the transcription and production of proteins that are important for development, such as axin, DKK2, and cyclin. Now, I did chemical exposures in order to perturb Wnt signaling, and the two main drugs that are used were LGK, which actually is a Wnt antagonist, and bio over here, which is a Wnt agonist. Now, Wnt signaling has been studied in zebrafish liver development. Basically, what we know is that Wnt signaling needs to be repressed for endoderm development, and actually it needs to be activated for hepatoblast specification and proliferation. However, the role of Wnt signaling in hepatobiliary differentiation and liver outgrowth is currently not well understood, which is the purpose of my project. So with that said, the exciting stuff, the results. So basically, I found that downregulation of Wnt signaling results in larger liver areas. So if you take a look at these beautiful pictures, you can see that compared to the control group over here, the 0.1 micromolar LGK actually has a greater liver area. And having quantified that, you can see that actually, yes, there's a 25% increase in liver area with Wnt signaling having been perturbed within those hours. And also, upregulation of Wnt signaling results in smaller liver areas. So these are surface projections of livers, and I calculated their volumes. And so right off the bat, again, you can see that the liver volume is significantly smaller with bio-treated zebrafish. And having quantified that, you can see that, yes, in the bio-treated zebrafish, their livers are significantly smaller. Now, one of the more interesting things I found is that the Wnt signaling not only affects liver size, but it also influences liver morphogenesis. Now, what does that mean? So let's look at these pictures. So this is a control group, and the magenta color represents hepatocytes, and the green color represents biliary epithelial cells. So you can look at the control group, and you see that the biliary epithelial cells are pretty well networked and dispersed in the liver. Whereas in the bio-treated fish, you sort of see this clustering of biliary epithelial cells, all sort of in one line, and not a lot of networking and branching. On the other hand, when you look at the LGK group, you can see that there is some networking, but also clustering perhaps around the edges of the liver. And so I tried to quantify this, and what I did was I looked at biliary surface area to liver volume, and I found no significant differences between the three groups. I also looked at the number of biliary epithelial cell nuclei compared to the liver volume. Again, not significant. So what that tells us is perhaps there's this pattern of clustering that's being influenced, that's being affected. And in order to quantify that, that was one of the questions is, okay, so how do you look at this? How do you study this? And so what this is every spot represents a biliary epithelial cell nuclei, right? So these are all the biliary epithelial nuclei that are basically have these spot projections. And so what the colors represent is that the blue represents these nuclei being very close to one another. So the closer they are, they're sort of in the purple range. And in the red range, it shows that these nuclei are actually further from all the other nuclei. So it's sort of a way to show clustering of cells by numbers and colors, so better visualizing it. And so if you take a look over here, you can see that the DMSO is pretty random and some clustering. In the LGK, you see clustering, but they're perhaps mostly around the edges. But I think the coolest one is bio, where you see all of these cells clustered together in one area. And I also, having quantified that, I actually found this to be significantly more clustered as compared to the DMSO group. And I would talk a lot more about the quantification and what that means. However, I will really come to my poster and I would love to talk to you about it. I would like to thank the Harvard Stem Cell Institute for giving me this incredible opportunity, and I would also like to thank the Gosling Lab, especially Wolfram and Scott, without whom this project really would not have been possible. Thank you so much for your time, and I'll be happy to take any questions.